in the last 5 years, with well, first we run these leptospirosis interventions, very large ones on Cuba, in a point that we were facing an epidemic, a dangerous epidemics in a region of Cuba, and then we use homeopathic remedies, also from circulating strain of leptospirosis to treat 2.4 million people. And unfortunately, we get very strong results regarding the control of the epidemic. So we managed to control the epidemic by using this treatment. And in addition to this leptospirosis trial, we start working with many other diseases like hepatitis A, dengue, influenza. And particularly with influenza, we just ran a very big trial comprising close to the 90% of the Cuban population. That should be 9 million people. And it's just finished last year, and we are just making the surveillance and getting data to see the impact of such interventions.
The research work is basically approaching a standard problem in lambing time, where we dock lambs' tails using a rubber ring. Now, quite clearly, this causes an element of distress for 20 to 30 minutes with the lambs. There isn't a sensible standard medical treatment for it, and if there was, it would be expensive and time-consuming. So my approach was to say, okay, well, let's try standard homeopathy, which we are told will work. So that I did, and after one pilot study and now a full randomized controlled trial, double-blind, well, actually triple-blind, technically, it's produced about a 30% reduction in the amount of pain and distress experienced by these little lambs.
Well, I don't do any basic research concerning homeopathy, but we do clinical research. We try to implement homeopathic treatment in the prevention of urinary tract infections. And as I'm not a homeopath, we work together with some very skilled homeopaths which do an additional treatment to the medical treatment that we do to prevent urinary tract infections. We try to stick to the concept of classical homeopathy, and for that reason we do an individualized treatment with just one highly potent or high potency treatment, and this is an individualized drug, so there is no drug to cure urinary tract infections. There are quite a few drugs which are tailored for the personality of the patient.
We've performed a lot of experiments with plants. We used a variety of different plant models, so we worked with plant cells, not so much, but especially with entire plants and various, especially ecotoxicological assays. So we worked with quails, with duckweed, with wheat, but also with apple trees and Arabidopsis thaliana and many other plant species, in order to make some sort of evaluation of different preclinical bioassays, either undisturbed or in a pathological condition. May it be stressed by an anorganic stressor like an arsenic poisoning, or by, let's say, some pathogens, some fungi, some bacteria that attack the plant, in order to see whether the plant will be able to struggle more efficiently with these external influencing factors when they are treated with homeopathic remedies. So it's an entire range of different preclinical bioassays based on plants, but we made also other experiments. And in total we examined 14 experimental models, starting from physicochemical investigations, touching microbiological bioassays, going over to cell cultures, up to the mentioned plant models.
Just now we've seen some interesting work, for instance, from the University of Strasbourg. These are not traditional homeopathic medicines. This is TNF, tumor necrosis factor, alpha tumor necrosis factor, highly diluted, has effects on release of interleukin-6. It's very technical, but this is relevant to things like rheumatoid arthritis, so very highly diluted. immune mediators have effects on cytokines. This is just one example. There's another area where there's correlation going on, gelsemium. A number of people have done work on gelsemium, both in animal experiments. It seems to have quite significant animal effects in animal experiments, standard experiments that are done, for instance, for tranquilizers. It seems to have similar effects to buspirone, standard tranquilizer, in high dilution. And we have a group from the University of Verona who are correlating that now with a high-tech method with looking at gene expression and have shown changes in gene expression. So this is now correlating animal findings with basic science findings.
One of the things that I've done is I've been involved in homeopathy research for 15, maybe 20 years. I was inspired to dive into research, but from the perspective of a practitioner who was into one aspect of homeopathy, provings. I was taught that provings were the science of homeopathy. And as I did more of these trials over the years, realised that the science doesn't stack up so well. And then after a number of years in Australia, where I was, after doing 10, 15, 20, 25 of these homeopathic provings over the years with multiple groups, with different trial designs, realised that we've got to tidy up this aspect of what we do. So, in answer to your question, what I was presenting here is a State of the Union address saying that for 200 years we've been doing something in homeopathy, and actually it stood the test of time, but only if you look at it from one perspective. Yeah? If you look at it from a purely scientific perspective and ask the question, is homeopathy a placebo response, then what we've been doing doesn't stack up. We've got to draw a line in the sand. We actually have to make some pretty hard decisions, and moving forward, our trials, our homeopathic provings, from that scientific question perspective, they've got to be better. And crucially, they've got to involve an ethics process. We can't escape that. This is human research, therefore it's got to go through, as far as I'm concerned, an ethics process. Even if it costs lots of money, we've got to do it. We've got to have that credibility. Our papers aren't going to be published in reputable journals unless they have that. And in addition to that, there's a number of specific issues of homeopathic provings, a bit of the nitty-gritty when it comes to when do you do things at what point in the specific trial that we need some agreement on. Now, that's happening already in various cells around the world. In the US, there's a group that get together and talk about that. There's the ECH group, there's the ECCH group in homeopathy that talk about that, and there's a number of people that have contributed to the literature. And we're getting there, and I think what I heard over the weekend was thanks for that. Good to hear, good to know where we're at, and we're on board, and let's talk.